All right, we're going to go ahead and get started. Thank you everybody for joining us this evening. For our next installment of the Young Scientist webinar series, we host this the second Tuesday of every month, October through April. And this evening we are hosting Megan Wilson, and she's going to talk to us about the Mortality Gauntlet, the early life stages of the Cabezon. And then our next month presenter will be on February 9th, Lisa Hildebrand, and she will be talking and sharing with us about investigating the fine-scale foraging ecology of gray whales in Port Orford, Oregon. A little bit about the Cape Perpetua Collaborative. My name is Tara Du Bois, and I'm the Communications Coordinator for the Cape Perpetua Collaborative. And our vision is to foster communication and collaboration within local communities. And our three guiding principles are community engagement, leveraging resources, and engaging in partnerships. We have a variety of founding partners, including federal agencies, state agencies, some local nonprofits, as well as the Confederated Tribes of Coos, Lower Umpqua, and Siuslaw Indians. And our focus is around outreach and education on the Cape Perpetua Marine Reserve. And it's Oregon's largest marine reserve. In addition, with its marine protected areas, has protected waters stretched from Yachats to Florence. And I like to share a fun fact every time we do one of these webinars. And today's fun fact is around the ODFW dashboard. They just launched this, I would say about a month ago. And it's a dashboard, it's a website that allows you to use a variety of different menus where you can navigate through the data that they have collected over the last 10 years. And it's across all of Oregon's marine reserves and their comparison areas. And you can find this dashboard at tinyurl.com/ODFW-data.app. And you can also find it on their website if you just do a Google search for Oregon Marine Reserves. And the Cape Perpetua Collaborative also hosts a variety of community science projects, as you can see here. Some of them are monthly, some of them are seasonal. And we also host a winter Cape Perpetua speaker series on Saturdays at 10 a.m., November through March. And you can find all of these events and webinars on our website at capeperpetuacollaborative.org. And just click on the events tab and that will take you to the calendar. And I also highly encourage you to connect with us on our Facebook page at the Cape Perpetua Marine Reserve. And we've also launched last year a YouTube channel for the Cape Perpetua Collaborative. And that's where we'll be hosting the recording of this presentation, as well as the presentations we have hosted in the past. And with that, I'm going to go ahead and introduce our speaker tonight, Megan Wilson. She is a PhD candidate in the Department of Integrative Biology at Oregon State University in the Plankton Ecology Lab. Her PhD research focuses on the early life stages of nearshore marine fishes in Oregon and how they grow and survive under changing ocean conditions. She completed a BS in Biological Sciences at California Polytechnic State University in San Luis Obispo, California, where she developed a passion for sustainable fisheries management and ocean conservation. In her free time, she enjoys gardening, knitting, backpacking segments of the Oregon Coast Trail, and learning to surf. And with that, Megan, I will turn it over to you. Stop share there. There you go. And while Megan is pulling up her screen, I just want to let you know that you are welcome. We're going to be doing our Q&A section at the end of her presentation, and you're welcome to add your questions as they come up throughout her presentation to the little Q&A box that you'll see at the bottom of your screen. Feel free to put them in there, and then we will get those answered. I hope you enjoy the presentation, and with that, Megan, I'll turn it over to you.
Thanks, Tara. Can I just confirm that you're seeing the PowerPoint?
Yes, we are seeing it. Looks great.
Okay, that's great. Thanks so much for that introduction. I'm so excited to have been invited by the Cape Perpetua Collaborative to talk a little bit about my research and some of my interests. I see some familiar names on the participant list and some folks I haven't met yet, and I wish that we could be doing this in person. But here we are. So thanks again for joining today. And the title of my talk is Survival Through Mortality Gauntlet, Insights about Population Fluctuation from the Earliest Life Stages of Marine Fishes. Before I talk too much about fishes, I want to talk a little bit about how I got here, and Tara mentioned some of this in my introduction, but ever since I was little, I grew up in Sacramento, California, and I've always had a really strong interest in the ocean and ocean critters. I think that picture, I'm probably about 3, and I believe I'm at the Monterey Bay Aquarium, getting my hands right in there with all the ocean life. And then I did my undergrad at Cal Poly San Luis Obispo, and I worked on this project, the California Collaborative Fisheries Research Program. And this is where I really developed an interest for marine fish populations. We did tagging studies to monitor where fish are moving and what their populations look like inside and outside of California's marine protected areas. And finally, I ended up in Oregon at the Plankton Ecology Lab for my PhD. And I shifted my focus a little bit to the early life stages of marine fishes. And I'll talk a little bit more about that shift as we move through the presentation. So we know that fish population sizes vary from year to year. In this figure, we see a bunch of different species in different colors, a couple of rockfish, lingcod. These are all fish that are off the Oregon and California coasts. And we see that these populations are going up and down from year to year. And we know that the populations are varying, and we know that these variations impact communities, economies, and ecosystems. For example, according to the FAO, about 3 billion people depend on seafood as their primary source of protein in the world. Here in Oregon, Oregon's commercial fishing industry alone accounts for over 6,000 jobs. And science tells us that healthy and diverse fish populations support resilient reef ecosystems. But new stressors are really challenging our understanding of these population fluctuations. So for example, many of us are familiar with habitat reduction impacting polar bear populations because their habitat, which is ice, is something we can see. But for many marine species, their habitat is restricted by tolerance for extreme temperatures, which is something we can't readily see. We can't just look at the ocean, know what temperature it is. We can't just look at a fish and know its thermal tolerances. But nevertheless, these habitat shifts and range contractions are occurring in marine systems as well. So, for example, in 2018, you might remember that fishermen here in Oregon landed more than 3,000 tons of market squid, shattering all previous records. And meanwhile, fewer squid were swimming off the waters of California, which is usually where you find them. And this is related to warming waters along North America. Because Oregon is generally cooler than California, the squid could survive in warmed Oregon waters, but not warmed California waters. So this is an example of that habitat contraction, similar to what we see in polar bears. And another way that stressors are changing our understanding of population fluctuations are these tightly fine-tuned rhythms in nature, tightly coupled by centuries of coevolution. So plants bloom in synchrony with the feeding periods of insects that pollinate them. But climate change can cause these plants to bloom or bees to reproduce out of season, decoupling that tight rhythm that they've developed. And even though we can't see it as well, these rhythms exist in the ocean as well. Climate change can cause plankton to bloom or fish to spawn out of season, putting tiny vulnerable offspring into the water column before there's enough of their plankton prey to sustain them. And so this really centers us on this question of why fish populations fluctuate. We know that fish populations are really important to people and to ecosystems. And we know that these patterns are changing in ways that we're only beginning to understand. And so in order to more comprehensively tackle this question throughout the talk, here are the 3 pieces that I'd like to bring in. So we'll take a brief history lesson to show us a little bit about how we've developed our understanding of fish population dynamics. We'll talk about some new and exciting tools after nearly a century of progress in our thinking. And finally, we'll talk about a study here on the Oregon coast that I'm really excited to share as part of my dissertation. So, okay, I want to bring us back to the past so that we can really understand how people thought about population fluctuation and how this thinking and these paradigms had ramifications for fishing communities and economies. So the year was 1902, and Northern Atlantic cod and herring catch that many European countries relied upon were at historic lows. People were starting to get nervous, and a bunch of scientists formed the International Council for the Exploration of the Sea. It's a multidisciplinary group seeking to understand how global fish populations fluctuate. And so this group was asking the very same question we're thinking about today. And the existing paradigm that they were coming into included the following things. So there was a species focus. And just to think about how this is different than how we think about fisheries nowadays, imagine if we didn't know that there were distinct populations of Chinook salmon, for example, that run in the spring versus run in the fall. If we just thought about it at a whole species level, Chinook salmon, we might miss a lot of those key differences in being able to predict what their populations will look like from year to year. In the paradigm, they believe that fish abundance was stable with constant renewal each year. So imagine how people who depend on harvest from the sea would behave if we assumed that a steady and constant number of fish were being added to the fishery each year. They did not know that overfishing was possible. They thought that there was an inexhaustible abundance of fish in the sea. And finally, they were operating under the assumption that year-to-year fluctuation was driven by fish migration. And I'm no fisherman, but I imagine that thinking the years when your catch was poor was just because the fish were constantly moving away from where you were might be a little frustrating. And so that committee that I spoke about convened and investigated for several years, and we see a paradigm shift. So I'll talk about that. So instead of a species focus, we realized that we need to be focusing on populations. So separate expectations for spring Chinook versus fall Chinook, things like that. Fish abundance was variable and renewal was irregular. And so you might imagine that fishermen or fisheries managers might change the way that they change their year-to-year behaviors with this assumption. And finally, or second to last, the paradigm shifted such that there was a finite abundance and overfishing was likely. And this last point that I've starred is the most interesting to me and most central to my research. And that is that year-to-year fluctuation is driven by recruitment variability, not migration. And I'll explain why this is so important. So most marine taxa have a complex life history. So take this Boccaccio, large-bodied rockfish. So bottom-associated adults spawn tiny larvae, which remain in the plankton community for weeks to months before returning to settle and grow in adult habitat. And this term recruitment here can be a little different depending on who you talk to, but how I'll define it in this talk is when a young fish, so those early stages in open water, survive through those early stages and begin to establish an adult nearshore habitat. And so the paradigm shift initiated by the ICES committee was saying that to understand why fish populations fluctuate, that big question that we're very interested in, you really need to be looking at the early life stages because those changes in recruitment are causing the year-to-year changes in the population. And so we now know that marine fishes experience upwards of 99% mortality during these early stages, and that's what's illustrated in this figure here. The y-axis represents population abundance, and the x-axis is days of life. And so we see that over a short period of early life, orders of magnitude of individuals can be lost, resulting in drastically different population sizes. So you can see this tiny change early on in life. You see the trajectory A and the trajectory B. That tiny change in early life is resulting in these orders of magnitude differences in the adult population sizes. And this is what we really care about, right? Because these are our dinner plate fish. And that's a difference in, you know, thousands of millions of fishes that reach the adult stage. And this 99% die-off can be thought of as a mortality gauntlet, which is in the title of my talk and what I'm going to talk a lot about today. And it's a gauntlet because it's all vulnerable young fish must pass through and very few survive. The nature of this gauntlet holds the secrets to understanding population fluctuation, and this is what's really fascinated me throughout my time at OSU. Johann Hjort, one of the scientists on that early committee in the early 1900s, developed a hypothesis that's being illustrated in this figure, And he suggested that the nature of the gauntlet is starvation. So you can see that gray box on the figure. It says critical period. And he suggested that thousands of individuals will not be able to navigate the transition from feeding on the yolk provided by their mother to foraging on their own on small plankton, that first feeding arrow. So according to this hypothesis, factors that regulate population dynamics might include things like the size of the yolk provided by the mother, or the number of small crustaceans available for larvae to forage upon, basically anything that's going to contribute to starvation during those early stages. But that's certainly not the only hypothesis out there. Hjort's hypothesis that we just talked about was published in 1914. So we've had over a century to develop hypotheses about the nature of the early life mortality gauntlet. And this is a similar figure to the one I was showing before, showing abundance decreasing drastically as fish age. And this figure is summarizing as of 1987, so a little bit outdated now, the various mechanisms of mortality that operate throughout a fish's life. So for example, physical processes like being swept away from suitable habitat by a storm or currents might be especially important for young larvae, who are weak swimmers, while predation may be more important more generally and throughout the life stages. Okay, and so our time turner up in the corner is bringing us back to present day. And we've had, like I said, a century of progress since the committee convened and had those major paradigm shifts. And so we've developed new tools and we're able to ask new questions. And so I'd like to spend the next couple minutes talking about some of the tools that the Plankton Ecology Lab, my lab at OSU, uses to answer some new types of questions. And so one of them is this instrument called the In Situ Ichthyoplankton Imaging System, or ISIS. This is a towed shadow graph imaging system. So we tow it through the water, and as we're doing that, it's taking these microscopic photos of the plankton community. And it's really interesting because we can capture images of rare things like larval fish, which are relatively rare in the big wide ocean. And also we can capture the abundances of more common things like larval fish's prey and predators. There's a pretty large field of view on this instrument, which is important for capturing things like the larger, like gelatinous planktons, kind of like jellyfish. You can imagine if you just dragged a net through the water, you might totally destroy any jellyfish that was in the water and you wouldn't know that it was there at all. But with ISIS, we can take images of them and capture that they were there. And this really allows us to ask more complicated questions about the predator and prey interactions in the water column, and ultimately how these interactions are shaping survival through that mortality gauntlet. And so I've been talking for quite a while now, and I want to give myself a break, and I want to give you all a chance to participate. So I've put up a couple different images that we've captured using ISIS on research cruises off the Oregon and California, Northern California coasts. And so you can think to yourself, make some guesses, or you can even put in the chat what you think that these little critters might be. And so here's the first one. I'll just leave it up for a couple seconds. Can I see the chat? Crabnoplii. Yeah, that's close. Yeah, this is a Crabzoea, crab larvae. Okay, let's do another one. Copepods. So this one's not a copepod. I wonder if that was the guess for the last one. A fish. Yeah, so this is a fish. This is actually a northern anchovy larva, really important small pelagic forage fish in our Oregon and California food chains. Okay, this is kind of a weird looking one. Does anybody know anything about this critter? Snail. Yeah, wow. This group is really on it. These are really interesting plankton. They're called sea butterflies. I'll put the name up here so you can see it. Or pteropods. They produce calcified shells, just like a snail. These organisms pull calcium carbonate, which are the building blocks for their shells, out of the water column. And so because of that, they're very sensitive to ocean acidification. When ocean waters are acidified, it's really hard to pull those building blocks out of the water column and really hard to build your shells. And so for this reason, these pteropods have been kind of like a canary in the coal mine for ocean acidification, because if you see a lot of these pteropods that are having really poorly constructed shells, it might be indicative of acidified waters. Okay, we'll just do one more. This one's so weird looking, I had to throw it in here. Let's see what people think. Oregon ocean plankton. Yes, it's not a fish, even though it looks like it has a tail. This is one of those gelatinous zooplankton that I was talking about that might be pretty challenging to find in a net, but we can capture it with these images. It's called a doliolid, say that one 10 times fast. And it's really fascinating. This is a colonial animal, so it's a group of colonial individuals living together. And this oval here is often called the nurse, because it carries developing individuals along that appendage that stretches away from it. Each of these little bumps on the appendage are individuals that might have different jobs. So some might attain food for the colony, some might be involved in reproduction, some might break off and live totally independently. So thanks for participating. I thought this was a really fun way to kind of show you what ICES can do and what kind of images we get, and talk about some cool critters. And also, I wanted to point out that this is actually how we generate some of our data for the lab. So we have a website, the Plankton Portal, and citizen scientists can do basically exactly what you did, help classify millions and millions of images. We're taking images with ICES, I don't know, every millisecond maybe. And so we've got tons and tons of images. And you'll see in this screen grab, there's one of the things that we just saw, a little doliolid. All right. But that's not the only tool we have. One of the tools that our lab uses is called the MOCNES, which stands for the Multiple Opening and Closing Net and Environmental Sampling or System Sampler. And this is a really interesting tool because if you imagine dragging a net from the bottom of the ocean to the top of the ocean, when you actually look at what you caught in that net, you don't really have any way of understanding if that critter was living on the surface of the ocean or at 100 meters depth. And so what the MOCNES does is from the ship, we can trigger the closing of nets at different depths. And so we send the net all the way to the bottom. And as we pull it up, we can say, oh, 50 feet, let's close a net. Oh, 25 feet, let's close another net. And so then when we sort through what's in the nets, we know what depth we caught it at. And it's an environmental system sampler because it has a plethora of environmental sensing tools. It can sense things like temperature, salinity, chlorophyll. And this is a really great coupled with the ICES because then we have the images, but we also have these biological samples. So you'll see in that picture, that other picture, you can see some larval fish in those petri dishes and some other gelatinous zooplankton. And then finally, I'll talk about a less high-tech, but still great tool that we have that I've used a lot for my dissertation, the Standard Monitoring Units for the Recruitment of Fishes, or SMRFs. We're really fond of our acronyms. And what this is kind of like a little fish hotel that's made out of garden fencing that we deploy just barely below the surface in nearshore waters. And these are really important data to collect because you imagine those huge imaging system, those huge nets that are probably as big as the walls of the room you're in. You cannot tow those near shore where there are wash rocks. But with these SMRFs, it only takes a small 20 or 30 foot boat to collect them. And the way we collect them is we send breath hold free divers off the edge of the boat and say, go get it. And we use a large net. We free dive down and use a large net to scoop them up. And then we shake out all the fish that were kind of staying in that little fish hotel. And a reason why it's so important to have these biological samples is because we can do otolith analysis. And otoliths are calcium carbonate ear stones that are in all bony fishes, so most of the fishes we see. And they're ear stones, kind of like our inner ear bones, and they help the fish with balance and orientation. And this is a really critical tool in fishery science that I will explain more in a little bit. But here I just want to show, this is an adult Cabezon, and its otoliths you can see are 5 or 7 millimeters big. So fishery scientists will extract those and use them for analysis. But of course, as the fish gets smaller, and the fish that I'm talking about today are, juvenile and larval, tiny, tiny fishes, the otoliths get smaller too. And so here's an example of a fish that I collected in the Smurf. Its otoliths are much smaller. And finally, here are some otoliths that I have collected from embryonic fishes, so fishes still in their eggs, or in their eggs, and larval fishes, so fishes that have just recently hatched from eggs. But you can see in those red circles faintly little otoliths. And just for some scale, we have a picture of a larval otolith circled in white. And there's a piece of dust under the microscope and an eyelash here for scale. And then we've got the juvenile otoliths, so the ones from the fish in the Smurfs, for scale compared to a little piece of rice. And so these things are very small, lots of microscope work. Okay, and the reason why fisheries scientists are so interested in these otoliths is because otoliths have growth rings, just like trees. So if you cut a cross section of a tree, you get a tree cookie. They've got rings, and you can see how old the tree was, how fast it was growing. And otoliths have the same type of rings. And so, if we were talking about the big adult fish with the half centimeter otoliths, we'd be looking at annual rings. But when you're looking at a lot smaller fishes, so larval and juvenile fishes, these rings are actually daily rings. And so they're depositing daily increments of material. So it's kind of amazing. It's a flight recorder. You can see exactly how fast the fish was growing at each day of life. Okay, so I'd like to pivot now to talk about a study that is the first part of my dissertation, and it's really near and dear to me in the Oregon coast. So the title of this part is The Tortoise and the Hare, Flexible Early Growth Strategies in Nearshore Fish and Recruitment Success in a Variable Environment. And I'd love to acknowledge my advisors, Kirsten and Sue, and my collaborators, the ODFW Marine Reserves Program, the Oregon Coast Aquarium. So we'll talk about Smurfs again. We use them in Oregon to sample those settlement stage juvenile fishes in the near shore. This is a multi-institution collaborative project. Like I said, we work really closely with ODFW Marine Reserves Program and the Oregon Coast Aquarium, and tons of OSU undergrad volunteers and grad students. And we sample bi-weekly from April through September, so every 2 weeks, free diving. So you can see in this picture here, a free diver is about to scoop up that Smurf with that big net and collect all the fishes that are inside it. This project has been going on for 8 years, and the project is ongoing. And we have sites, we have Smurfs located in and around the Otter Rock Marine Reserve and in and around Redfish Rocks Marine Reserve. But for the purposes of this talk, I'll be talking about the samples that we've collected only in our Central Oregon sites, which are in and around Otter Rock Marine Reserve. And like the title of my talk gave away, I'll be focusing on Cabezon. We collect several different species in the Smurfs, rockfish, greenling, snailfish, and Cabezon. So Cabezon is a commercially and recreationally important fisheries species. And previous work in our lab has shown that they recruit to nearshore kelp forests over an extended period of time, arriving in nearly every month sampled, so April through September. This multiple pull strategy, or constant coming in strategy, is unique because many of the other species we collect in the Smurfs, including things like rockfish, they only arrive over a short window. So like maybe only in the month of July, whereas Cabezon are coming in this whole time. And just to give us a little bit more context, the Northern California current, which is the large marine ecosystem that Oregon is a part of, is dynamic at multiple time scales. And so this is a picture of Redfish Rocks Marine Reserve, one of our study sites. And those of you who are from Oregon know that the ocean looks like this maybe 7 days a year. Certainly doesn't look like this right now. And there's tons, and this all to say, there's tons of variability. Monthly scale, we can have storms, we can have upwelling regimes. At a yearly scale, things like the marine heat wave that just came through in 2014, 2016, or more cyclical cycles like El Nino. And even on the decadal scale, there are cycles that fluctuate every decade and change things like temperature and productivity in the ocean. So we've established that Cabezon exhibit an extended recruitment window. They're here through April through September. So they're out in the open ocean for quite a while. And we've also established that the Northern California current is highly variable. So it follows that Cabezon experience a vast range of environmental conditions during their early life. Cabezon's unique recruitment strategy affords us the opportunity to examine environmental effects on early life at multiple time scales. And so we hypothesize that Cabezon will exhibit a diverse portfolio of early life history traits across the recruitment season. And this portfolio that I'm talking about is the same idea as an investment portfolio. In an unpredictable environment, the strong portfolio is a diverse one. And we know that Cabezon are inhabiting a really dynamic environment. So they probably have a diversity in tools to deal with all this variation. So to test this hypothesis, we use otolith analysis, like I talked about before, to examine early life history traits from 5 years and 3 groups within each year. So 2013, 15, 16, 18, 19. And then within each of those years, we look at May, June, and then lumped together July, August, September. Okay, here's this photo again. Sorry about the auto advancing here. Early life history traits, which is what we're going to be using to look at this like portfolio effect, are measurable characteristics that determine how a fish navigates through its early life. So we know that early life is when the mortality gauntlet occurs. So understanding how variable these traits are clues us into what kind of strategies are being used to survive through that gauntlet. So now that you've seen, oh, here we go. And so we can measure a number of traits like the age or the number of days spent in the plankton. And that's just counting up all of those increments to the very edge of the otolith. We've got growth rate. So growth rate would be just the space in between, the width between 2 consecutive increments. And you can also use otolith size as a proxy for fish size. And so, So for example, if you go out to ring number 45, you can see how large the fish was at day 45. And so now that you've seen how we measure these traits in otoliths, I'll show you the data in some summary figures that help us compare these traits between months and years. Okay, so here's an example of some of that data we can get from the otoliths. We have plotted here, so all the different years, 2013, 15, 16, 18, and 19. We've got age at settlement on the y-axis, and so that was that yellow line where we count up all the rings until the edge. And then we've got month in different colors. So July through September's in blue, June's in green, May's in pink. Oh, geez. Okay. So in some years, I just want to draw your attention to some of the variability in these traits. In some years, like in 2019, fish spend about the same time in the plankton, so about 110 to 120 days. But if you take a look at a year like 2016, there's a much larger range. The individuals that arrived early in May and June were about 110 to 120 days old, but the ones that arrived in July, August, September only spent 85 days out there in the open ocean. And this is really curious. Imagine our surprise when a human baby reaches a major developmental milestone like talking or walking, or in a baby fish's case, settling in adult habitat, a whole month earlier or later than we expected. We, of course, wanted to know how and why this was happening. So we looked at another early life history trait that we talked about before. Growth rate, which comes from the width between consecutive increments on the otolith, Fast growers are spending less time in the plankton than slow growers. So you can see those fast growers far to the right on the x-axis have much lower or younger than the ones that were growing slower, which are closer to the origin. And so growth rate and age could have counteracting effects on size. For example, they could be growing really, really fast, but not spending very many days growing, or they could be growing really slowly for many, many, many days. So you can imagine that this could potentially result in consistent size at settlement across months and years. So that's what we looked at next. And that's not exactly what we saw. Size at settlement varies across months and years. And it's pretty intuitive that size for a young fish is really important when it arrives in adult habitat. It's really important for survival. Basically, the bigger you are when you arrive in the adult habitat, the less things can eat you. And we see again, some years like 2016 with drastically different sizes at settlement, almost 20 millimeters different based on when they arrive. So the early ones red and green and the later ones in blue. And then we see some years like 2015 where the difference is a lot more minimal. All right, and so we wanted to take a deeper dive into the analysis of growth by looking at the average growth rate on each day of life. And so we have age on the x-axis, so that's how old they are, and then on the y-axis, we have the growth rate. So remember, that's those increment widths as you move along the otolith towards the edge. And I really want to draw your attention here to the blue line, which represents July through September months, those late settlers, and it's consistently diverging from the other lines. So in all 5 years, you can see that pink and green line, those early settlers, is below the blue line, those late settlers. So they're growing quite a bit slower. All right, and so I know looking at all these different colored lines can be a little dizzying and kind of abstract, so I'll share a fun analogy before we move on to our next figure, which is quite similar. So this is me and my cousin. He was born in May and I was born in June of the same year, so an early settler and a late settler, if you will. Imagine if you took a height measurement each day of our lives from birth until present. You might see that at a given age, so when we were, I don't know, maybe 4, you took a height measurement. You can see that I was taller at that age. Then you can take another height measurement at our next stage. I was a little taller still at that age. And following all the way through our lives, you can see that for a while I was a little bit taller, but eventually he overtook me and is taller than me today. So at any given age, you take the size of an individual. And that's what we're looking at here. On the y-axis, we have size, and on the x-axis is age. So at any point in the figure tells you size at a given age. And I want to call your attention to those blue lines again. We see the exact same pattern as the last figure we looked at, where the early settlers are in pink and green, and they're growing slower and are smaller at a given age than those late settlers in blue. You can see in some years like 2019, the blue line remains elevated for the entire period, so the whole time. In some years like 2016, the pink and green lines eventually catch up to the blue lines, kind of like my cousin did with me. Okay, so, sorry. We've documented variability in several early life history traits now: size, growth rate, age. And we want to return to our big overarching question that we opened this whole talk with. How do these characteristics impact survival through the early life mortality gauntlet? And ultimately, what does that mean for population fluctuations from year to year? And so what we found, well, that's a big question, first of all. But so far we found that one trait, early growth, is indeed related to recruitment rate. And that's what I'll show you here. Meaning that events that occur in early life and affect growth have an effect on the number of fishes returning to the nearshore adult habitat. And that's what we're measuring by recruitment. And so they're related. What you experience in your early life changes how many of you show up, and that has ramifications for adult populations. But I will say, with the relationship we have here, we're faster early growth, farther along the x-axis, is associated with lower recruitment rate, so fewer fish arriving total. That's not exactly what we expected. But we have some ideas about why that might have been. In a warm year, we know that fish metabolism is directly linked to temperature. So we might expect that in a warm year, the fish might be growing faster. We know that faster growers spend less time in the plankton. Oh, we showed that earlier. And maybe that means that fewer fall victim to the mortality gauntlet. But that's not really what we see here. But this could be because we also know in warm years, they might be growing faster, but there might be fewer high quality prey items for larval fishes. We know that in warm years, there's just not enough of those really fatty, delicious copepods that larval fish love to feed on. So perhaps that boost in growth rate from the higher metabolism from the warm temperatures is essentially canceled out by a lack of food to sustain that fast growth. And so this is really what I'm going to be looking at in my next steps for this project, which is including temperature and prey data to see, figure out why we're seeing this kind of curious pattern. All right, to sum up, here are some of the things we know. We know that there are these nearshore adult cabezon, they're laying eggs. Their larvae are hatching out of those eggs. They're going offshore to the open waters. And then they're returning to the nearshore. We catch them in the smurfs as juveniles. And we know that they're growing, and their growth rate has to do with recruitment. So it has to do with how many fish are coming in and also what size they're coming in. So that's what we've established, that early growth rate is really important for recruitment. But there's a lot of things that we still don't know. And that's what I'll try and capture here. So we don't necessarily know how food availability, temperature, and the predator field, so the jellyfish that might eat that larval fish, how do those things interact to impact growth? And then how does that change how many fish are actually showing up in the near shore and making it to this next juvenile life stage? Similarly, or in parallel, we don't know how the prey field, the temperature, and the predator field are impacting how fast these juvenile fishes grow and how many make it past and can replenish the adult population. And so those are some of the things that I am focusing on in the next steps of my PhD. So what are some important sources of mortality for those younger larval cabezons? So is it temperature, food, is it predators? That's some work that I'm doing in collaboration with the Northwest Fisheries Science Center at Hatfield Marine Science Center. And also so the other side of that schematic that I showed before, how does early growth affect survival to the older tide pool cabezon stage, and what kind of factors are important for mortality there? This is a really cool project that I'm very excited about because it takes place in and around the Cape Perpetua Marine Reserve, and it allows me to bring together a lot of friends and citizen scientists and BioBlitz members and undergraduates, and we go out collecting juvenile cabezon in the tide pools. All right, and with that, I will say that if this was something that was really interesting to you, my lab mate, Dr. Will Fenney, who just defended his dissertation, will be giving a talk about some similar concepts, but in black rockfish rather than cabezon. So that's a picture of Will and I. We went out smurfing several times together, and it was always a blast. And I'll also say, I just want to give a shout out to everybody on this list, the Smurf volunteers, Smurf collaborators, my lab mates, funding sources. Without all of these people, these types of projects wouldn't have been possible. And also feel free to follow me on Twitter or contact me. And with that, hopefully, I should be able to take any questions.
Fascinating research. So we did have a question come in, and if you have any questions, please feel free to go ahead and plug them in the Q&A section. All right, so our first question, are you taking into account or considering the coastal zone's connectivity related to the nursery grounds?
That's a good question. So yeah, we can definitely consider those kelp forest habitats that the Smurfs are emulating as kind of nursery grounds for those young fishes. And I'm assuming by connectivity to the nursery grounds, you mean like the offshore larvae coming into the nursery grounds. And that is a big assumption in our work. And I haven't explicitly addressed that, no. It might require some tools, maybe genetic testing of the larvae that are offshore and then the juveniles that we find in the Smurfs in those nursery grounds to see if they're actually coming from the offshore directly onshore, or are we actually getting larvae from California that are brought up by currents and then come into Oregon's nearshore? So that's a really big question for a lot of different marine species, and we really just don't know without doing some of that genetic work.
Okay, next, when do the rings of the otoliths transition from being daily rings to annual rings, and how can you tell when it changes?
Oh, that's a good question. I talked a lot about those annual rings, but I haven't actually read annual rings. All my work has been done with the daily rings. And so we know that fish are depositing rings daily. It's just that once they get so old, those rings kind of start to squish. And so we see kind of like a darker and lighter banding that has to do with year rather than day. So it's not necessarily that they stop depositing daily rings, it's just that they start to get so squished. And I would say the oldest fish I've looked at were about 270 days old, so a little bit younger than a year, and it was starting to get pretty challenging to distinguish the daily rings. So I would say probably around a year is when you would want to start looking at annual rings instead of daily.
Are you accepting assistance for the upcoming project from other biologists?
Ooh, we are always looking for assistance. COVID has changed all of the field work, but assuming that it's safe to do field work this summer, we're always looking for assistance to help jump in the water with us and collect the Smurfs from the near shore on boats. But then also, I'm going to be probably looking for assistance to help out with doing the tide pool collections, which is a little bit more accessible, just dragging a net along the tide pools.
And is the best way for them just to contact you directly?
Yeah, definitely. I might not have an answer right now. Nobody's really sure what's going on this summer, but I'll definitely keep in touch.
And then why are July, August, and September fish lumped together?
Oh, that's a good question. Mostly because of sample size. So as the Cabezon are coming in from April through September, but they start to kind of trickle in July, August, and September. And so in order to make any conclusions about like significant or marked differences in size or growth rate, I needed to lump July, August, and September together. Basically, my criteria is that I need 20 fish in each group. And so sometimes I'll have 10 in July, 5 in August, and 4 in September. So I have to lump those together. If I had more samples, it would be great to really tease them apart and see if, for example, the ones that come in really late in September are just like growing through the roof.
How long do Cabezons spend in each life stage? For example, pelagic, Smurf, etc.
Oh, that's a good question too. So I actually recently did a small experiment where I was rearing Cabezon eggs. And so it appears that they're in their eggs, so being incubated for between 2 and 3 weeks. And then they hatch, and then they're in the open water column as larvae for anywhere from 3 to 4 months. And that's actually exactly the figure that I showed that had age broken up by months. And so the age that they arrived in the Smurf tells me how long they were larvae. So, and we got anywhere between 85 and 120 days. That's how long they're larvae. And then I'm not sure how long it takes for them to leave like the kelp forest and move inshore to the tide pools. But I think it's really variable because some of the tide pool fish I collect are about the same size as the Smurf fish and some are much bigger. So I think it really varies from individual to individual. And then I guess they spend, I think, I want to say about 3 years growing into a size that is harvestable by the commercial and recreational fisheries.
And then it looks like we don't have any other questions from our audience at this time, but I want to end with one. I always like to ask, what was the inspiration for you behind why you started doing this research?
Yeah, that's a great question. I think it all started when I was an undergrad and I was on a fishing vessel. We were doing that tagging study where we hook and line catch these adult fishes and we tag them and we can measure their home ranges and we can see how their populations are fluctuating. And I just had all these questions that I couldn't answer by just looking at the adults. And so I wanted to know, like, where are they coming from? Like, what are the larvae doing while they're offshore? How do we know how many are going to come back the next year? And kind of like I walked us through in the beginning of this talk, to answer those questions, there's really not many other options besides looking at the early life stages. And I love being in the water, being on boats, free diving. So this is perfect.
That is perfect. I appreciate that. So that is a good little kind of announcement we can say. So get out there and volunteer. You never know where it might take you, huh?
Yeah, definitely. We've had some really excellent volunteers on our crew. We've had undergrads, we've had high school students.
Yeah,
that's great.
Great. And then just one last question. Can you give your email again? We've got a question here about what the best way to contact you is.
Let me put my email in the chat and then I will also put my Twitter handle if anybody's on Twitter. I'll make sure I spell it right. Okay, that's a good way to get my attention too.
Perfect. So it's Wilso, W-I-L-S-O, Meg @oregonstate.edu. And then her Twitter handle is @WilsonMeg Wilsomegalodon.
Yeah.
Very cool. And with that, Megan, I just want to thank you so much for being with us tonight, especially as you're teaching a class and everything too. I really appreciate that. And everybody, this will be recorded and posted on our YouTube channel, so you can refer back to it or share it. And with that, I hope you all have a wonderful evening and a wonderful week. Have a great night. Bye-bye.